All right, hello everyone. Welcome to VSPG. Today we're hearing from Dr. Susanna Porter from the University of California in Santa Barbara. And Susanna will be talking about early eukaryote diversity, a review and reinterpretation. Jim is going to introduce Susanna for us today. Please go ahead, Jim.
Hi, everyone. It is my pleasure to introduce this week's VSPG speaker, Dr. Susanna Porter from the University of California, Santa Barbara. I've had the good fortune of rafting down the Grand Canyon with Susanna and can personally vouch that she's just as awesome in the field and on the river as in the lab and classroom. Susanna earned her PhD in biology from Harvard University, where she worked with Andrew Knoll, and she holds a BA in mathematics from Yale. Since then, she's become one of the most influential voices in Precambrian paleobiology, known for her thoughtful, rigorous, and often bold re-examinations of long-held ideas. She is a full professor at UCSB, a recent fellow of the Geological Society of America and the Paleontological Society, and an award-winning mentor and teacher. Her work has transformed our understanding of early eukaryotic life, biomineralization, and the environmental context of early evolution, with fieldwork that spans from everywhere from Arizona to Australia. Please join me in welcoming Dr. Susanna Porter.
Thank you. Thanks so much, James and Alex, for organizing this. And thanks, everybody, for showing up. I'm excited to talk to you today about a paper that was just published in Paleobiology as part of their 50th anniversary issue. I have here the cover of the very first issue of Paleobiology that came out in winter 1975. And I don't know if you can read those names there, but it's a lot of the big thinkers in paleobiology: Stephen Jay Gould, Steve Stanley, Dave Raup, and so on. And this journal represented a sort of a turning point in thinking about paleontology, focusing on on fossils and the way what they can tell us about the evolution of life, rather than just fossils being useful for biostratigraphy. This was the journal that made me decide to become a paleontologist way back when I was starting out grad school, and it was the first journal I published in, and so this actually has a personal importance to me. And so when I was asked to contribute a paper to their 50th issue, I said yes, but the topic that they wanted me to talk about was geobiology. They wanted me to write a 8,000 word review on the topic of geobiology, which I thought was a little overwhelming. I realized what they really wanted me to talk about was early life. Again, that's still too big. And so what I decided to do was to narrow down the topic and just focus on early eukaryote diversity. And I thought I figured I'd just sort of do an updated review of what was going on with the early fossil record of eukaryotes. And there were two reasons that I wanted to do that. One is that I thought it was fitting for this journal at the time that this first issue came out 50 years ago. There was basically no Precambrian record of eukaryotes. I mean, the sort of widely accepted earliest evidence for eukaryotes was latest Ediacaran in age when this issue came out. So I thought it would be interesting to see how our knowledge of early eukaryote diversity has changed over the last 50 years. And then the second reason that I thought it would be timely to do this is that Leanne Reedman, my sort of main collaborator on this project, had been building a data set as part of her PhD work. She published a paper with Pete Sadler using CONOP, looking at the pattern through the Tonian, and she had continued to build that data set. And this was a beautiful, carefully vetted data set on early eukaryote fossil occurrences. And so we thought that this would be a good time to look at the patterns in that data set. I brought in a couple additional collaborators, my former PhD student, Tina Woltz, who is now a research fellow at Imperial College London, David Gould, a professor at UC Davis, who's an expert on the molecular fossil record of eukaryotes in the Precambrian, and then Jay Kellogg, who I went to grad school with. He is a geophysicist, now a physics and computer science teacher at a boarding school outside Santa Barbara, and he also happens to be my husband. So useful to have a collaborator in the house. So I thought this would just be sort of an interesting update on the state of affairs, and it turned out to be partly prompted by some great reviews I got from Nick Butterfield and Shuhai Xiao, to be a really surprising result that was really unexpected. So I'm excited to talk to you guys about this today. So just so we're all on the same page, eukaryotes, you're familiar with the animals, plants, and fungi, and seaweeds. But of course, it encompasses a much larger, diverse group of organisms, and primarily is dominated by these single-celled organisms, which are often called protists. So if you're looking at the Precambrian fossil record, you are mostly dealing with these single-celled organisms, the protists. And most of the fossil record of the Precambrian eukaryotes is made up in these organic walled microfossils, OWMs, in shale. And you can see this, it's basically the left half of the screen. We do have some larger macroscopic fossils, known as carbonaceous compressions. You can see this example here in the middle. And also there are three-dimensionally preserved fossils in chert. And then I highlight these two groups just because they represent actually a significant amount of Precambrian diversity for their time. The apatitic scale microfossils shown up top here, and the which are thought to be scales, maybe that surrounded a single cell, kind of like coccoliths today, coccolith phorids. And then these vase-shaped microfossils, which are thought to be the little tests that were formed by an amoebae. So the traditional model of early eukaryote history has posed something of a conundrum. And that is because though eukaryotes appear, we think, at least 1,700 million years ago, and maybe even earlier, if Gripania spiralis is a eukaryote, but at least by 1650 million years ago, they appear to have remained low in diversity, marginalized members of bacterial-dominated ecosystems for hundreds of millions of years, basically like half of their reign on Earth. And it wasn't until the mid-Tonian, 800 million years ago, that they underwent a dramatic diversification and ecological expansion. So that's been the model. And this model is based first and foremost on the record of eukaryote diversity, those microfossils that I showed you, mostly microfossils, but also macrofossils, that you can see here in this example from Knoll, 1994, showing an increase rising up through the Tonian. And then it's also corroborated by the record of molecular biomarkers, which I've indicated here by these circles, the fact that we don't have any eukaryotic steranes through the Mesoproterozoic or Paleoproterozoic, and it's not, even though we have other biomarkers in these assemblages. And it's not until around 820 million years ago that we see these eukaryotic steranes show up. And of course, some of you are aware that Jochen Brooks has a paper that changes this story. But this has been the model for many years. It's also corroborated by the fact that we have a couple of crown group fossils appearing around this time. It all seems to make sense that this is this time of radiation. And then those special groups, the apatitic scale microfossils that are so diverse, and the vase-shaped microfossils, there are these sort of biological innovations showing up right around this time of this great diversification. So, this seems like a reliable story, and it is one that is been repeated, over and over again in reviews of early eukaryote evolution by a whole number of Precambrian paleontologists, including me. So as a result, this has driven a lot of other Precambrian researchers, geochemists, for example, to try to understand what is going on at this time in the Tonian. And I just have a sampling of different papers here representing a whole slew of papers that are out there trying to sort of solve this interesting conundrum, why there's been this boring billion in eukaryote evolution and then this flowering in the Tonian. Okay, so this is now where I turn to our paper, where we reviewed studies of early eukaryote diversity over time. So what's interesting is that you can see that the evidence for this Tonian radiation of eukaryotes shows up in these very first compilations. So the first one, published in 1982 in Nature by Vidal and Knoll, it's just based on a handful of assemblages. This is Grayson Shale way back there, that little dot. And then sort of, you could see we don't have any data through this time. And then there's that peak at 800, 700 million years ago. Schopf in 1992 undertook a really extensive analysis based on 2,800 fossil occurrences from hundreds of formations spanning 2,500 million years to 550 million years ago, looking at a lot of different ways of measuring diversity, but they all show this same pattern, this significant peak in the Tonian. So here we have Andy Knoll and Bill Schopf agreeing on something. It must be real. Then Knoll took a more selective approach here. He just looked at 15 fossil assemblages from the pre-cryogenian interval for that billion years. And the thinking here was that the patterns were significant enough that you could just look at a few of the sort of really representative high diversity assemblages through this time and get a picture of what's going on. And in fact, I grabbed this quote from that paper. He writes, "It is unlikely that any combination of sampling, taxonomic, or geochronometric errors exerts a major control on the principal pattern revealed by this analysis." In other words, sure, it's going to be wrong in the details, but the big pattern, the big picture that diversity is low in the preceding billion years, Paleo and Mesoproterozoic, and then there's a big diversification in the Tonian, that is real. In the 2000s, there was a shift to looking at within-assemblage diversity instead of global diversity as a way of controlling for sampling. And so here, each one of these boxes on the left in the Knoll et al. 2006 represents a single assemblage, and the width of those boxes represents the uncertainty in the age of that unit. There's an updated version here, Cohen and McDonald, as well. Again, sort of limited numbers. The Knoll et al. Is based on 17 fossil assemblages, but you can see that rise up to 800 million years ago. Cohen and McDonald did a lot more statistical analyses, had a lot more data available to them 10 years later. And they concluded that mean within assemblage diversity does increase through the Proterozoic, and it's driven by the fact that we get a lot more high diversity assemblages in the later part of this interval and around the Tonian. Okay, so these studies seem to come out every 10 years or so. So here we are 10 years later after Cohen and McDonald. And so we decided to put together our own analysis based on the fact in part that there's a lot of new data that have come out in the intervening 10 years. And as I said, this data set was largely built by Leanne Reedman. We're covering the period from 1900 million years ago to 700 million years ago. It comprises 713 species occurrences. We have 145 formations. We decided to sort of in the past, the previous analyses didn't really standardize. Sometimes it was a group, sometimes it was a formation. And yes, I realize, not all formations are necessarily equal, but it's the best we could do to try to standardize this. So we have 145 formations. We did decide to split up shale-hosted versus chert or carbonate-hosted assemblages, and by chert, often it was silicified carbonate. So we put those into one category and the shale into another category. And so we have more assemblages than formations as a result, because some formations have both. you can see there's a lot more shale-hosted fossil assemblages than chert. There were 182 species, and we took special care to divide those into species that we thought were likely eukaryotes, sort of based on widely accepted characteristics. And then another 32 that we thought were possible eukaryotes, but there was uncertainty in the literature about whether they might be prokaryotes. It turns out that, as you'll see, this did not make a difference at all to the analysis. And so I think for most of the graphs I'm going to show from here on out, we're just going to show the entire group, the 182 species, and not worry about dividing up the two, though in our paper we do divide them up. I think this is really important. The taxonomic assignments were vetted and updated. So this was not a simple scrape, you know, like AI scrape through the literature. This was Leanne very carefully examining all of these papers, seeing if the claim of that species held up to scrutiny based on the images that were provided. And then she also updated that taxonomic assignments based on, you know, the latest thinking about, oh, that species is not part of that, that's actually in that species, and so on. So this is a very carefully curated data set. 713 species, as I said, from 276 papers. And then the other point to make is we decided not to include these simple laeospherids. These are these simple smooth-walled acrotarchs that are sort of have an artificial classification, and who knows what they are. Some of them might be prokaryotes. They're just sort of too artificial to deal with. So we didn't include those. So first, we were curious to see how our compilation compared with the most recent compilation of global diversity that is available, and that's from Knoll, 1994, 30 years ago. So here's Knoll, and you can see the time bins are not equal, but that's just based on sort of uncertainties about ages. And here are our results. So you see there's a significant difference, and in particular in this early Mesoproterozoic and late Paleoproterozoic time. And that reflects the fact that there's been a lot of focused study in the last 30 years, and in particular last 10 years or so, on this time interval. And so we found a lot, we've increased the diversity. And here's one example. This is these beautiful fossils that Leanne has described from the 1650 to 700 million year old Lumbunya group in Australia. It shows quite a significant eukaryote diversity. The other thing I wanted to point out is that that big spike there at 800, just before 800 million years ago, a lot of that is due to the apatitic scale microfossils. There are 38 species of these described, but I think it's important to note that these are from a single locality, a single formation in the Yukon. And because there are so many species there, they really kind of skew the pattern. So it's sort of like adding the Burgess Shale, you know, into a compilation of Cambrian diversity and seeing that there's a peak there, right? So here is an analysis where we just standardize the bin sizes. So 200 million year bin sizes. Now we have a better resolution on the ages, so we don't have to resort to the larger bin sizes. I removed the apatitic scale microfossils from this analysis. And it's interesting, you see, you know, there's a big increase into the Paleoproterozoic and Mesoproterozoic, and then there's that big bump into the Tonian. But the boundaries of these bins, right, of course, have an effect on the pattern where you put the boundaries. And so we decided to use a moving window to look at this pattern. So we have this 200 million year moving window, and we calculate, you know, species diversity, and then we shift it over 20 million years, calculate it again, shift it over 20 million years, and so on. And so this is what this averaging looks like. Again, you see this peak in the Tonian. Here are the results if we do include the ASMs, the apatitic scale microfossils. They have a significant impact there on the diversity. And then just to show you, I said if we just look at likely eukaryotes, remove the possible, the uncertain eukaryotes, that's what the curve looks like. So it's lower down, but it's really not telling a significantly different story. Okay, so one thing we were concerned about and our reviewers, Nick and Shuhai raised, was the question of sampling. What about the effects of sampling? We know, for example, studies with modern diversity, and here's one I just grabbed off the internet. This is this paper looking at sponge diversity in the Eastern Mediterranean. And of course, how many samples you grab from a particular place. So here they have, different areas of collection, north, middle, south. How many samples you grab is going to obviously affect how many species you find. And so a typical thing that ecologists will do is rarefaction curve. So this is where you're subsampling your data set, reconstructing it, calculating the average, and for, if you were to have fewer samples, how many species would you expect to find, and so on. So I wanted to do something like this, but the problem is that in our data sets, not all sampling is equal. So this is an example from the Tonian. What I've shown here is on the y-axis, it's that within assemblage diversity. Every one of those bars is an individual assemblage, and I've just rank ordered them there along the x-axis, right? So we have almost 60 different assemblages, fossil assemblages from the Tonian. One of them is, 19 species, a couple of them are 18 species. These are really monographic treatments. I mean, one of these is like Butterfield et al., 1994, this monograph from Svalbard. One of them is the vase-shaped microfossils, the Chuar group. But a lot of these papers are first discovery reports. They're like just a quick pass, found fossils here. And so it didn't make sense, I think, to sort of just average them, right? To subsample and then average real monographic treatments with just a quick, here I found a fossil. And to illustrate, this sort of process of paleontological study, I think this is a great example. This is Tina has a paper that is in prep where she went back to look at the Uinta Mountain Group from Utah, 750 million years old. And at the time, there were 2 species that had been described. And her work yielded the discovery of another 14 species. So just this sort of more intense effort really has an effect on what the diversity is. So our group, we talked for a while about how to get around this. Maybe we should weight some and then some analyses more than others and do the rarefaction of sort of these complicated ideas. So that's where I roped in my husband and I said, could you help us? You're this computer science whiz. Could you help us come up with a program where we could do some subsampling, et cetera, et cetera? And the brilliant mind that he is, he's like, oh, you're just making it too complicated. Instead, let's just look at the number of formations in your data set and have first pass and see how that compares. And so I said, oh, that's a good idea. Okay. So here we have species diversity there. You've seen that graph before. And here was the graph of the number of formations in our data set. And we sort of gasped. We're like, oh my God, those look pretty similar. And he said, yeah, let's graph them, X versus Y, number of species, number of formations. So here we have X, number of formations. Here we have Y, number of species, R squared, 0.97. So then we thought, oh, well, just to continue with this, residual number of species, right? So this is how far off of that perfect line, of that line each dot is. You can see the residuals are quite small. I mean, 1 or 2 species, just noise. If you include the ASMs, the R squared is 0.99. We tried to break this pattern. We're like, okay, well, let's just look at the shale window, not the chert. There are no ASMs in the chert, in the shale. So they're not an issue. If we just look at shales, which is where most of the assemblages are, R squared 0.98. if we just look at 100 million year bins, we prefer 200 million year bins because the sort of range of values of ages for a lot of these formations, the median is 150 million years. So we thought to sort of capture that at 200 million year bins made sense. But if we look at 100 million year bins here, it's messier, but the R squared is still really high, 0.87. You can see maybe the residuals are suggesting maybe there could be a Tonian, something going on in the Tonian, but I hesitate to make a big deal out of it. Here's the pattern with that moving window. You've seen that one before with the number of species. If we graph on the number of formations, there's a pretty striking correlation there. Looking at the moving window now and doing that same XY plot, and you can see the ages here are color-coded, the different dots. So we have some Tonian that are down here and so on. R squared, 0.93. So we were sort of couldn't believe this. And we tried to figure out, is there something going on with the program? Are we thinking about this in the wrong way? But we couldn't escape the fact that there seems to be an extraordinarily strong correlation between the number of formations in our data set and the number of species. Now, if you recall, I talked about how there was a shift in the 2000s to focusing on within-assemblage diversity instead of global diversity as a way of controlling for sampling. The Knoll and the Cohen et al. Papers in particular. But the problem with this measure of diversity through time is that if you look at these graphs, your eyes are really focused on the maximum within assemblage diversity. And this is what these, Knoll in particular, they picked for these units, right? They picked the assemblages that were most diverse for their time. Cohen and McDonald picked some lower diversity ones, though, as well. But your eye kind of tracks that max. And if you graph the max within assemblage diversity from our data set, this moving window, and you plot that versus the number of formations from that particular window, that also is correlated, the sort of, the lowest value there. You can see there's this sort of whole space that is not filled. And that makes sense because, the more formations you study, the more likely you are to hit a bigger and bigger jackpot in a particular time period. So I guess we're left with this conclusion here based on these graphs that at least in our data set, the best predictor of diversity in any given time bin is just simply the number of formations in that time bin, the number of formations that have been studied by paleontologists, and they've found eukaryotes in them. All right, so anytime you have a correlation, right, you have to ask, what's the causality? And we're putting aside total random coincidence as an explanation. There's three explanations. There's either Y is driving X is driving Y, or there's a hidden variable, Z, common cause, that is driving X and Y, right? So what does this mean in terms of thinking about eukaryotes? Y driving X, that is saying that the diversity of eukaryotes is driving the number of eukaryote-bearing formations in our data set. Remember, all of the formations in our data set are, of course, eukaryote-bearing, right? So it's possible that what's going on here is that this correlation reflects the fact that there is a real Tonian radiation, and that as eukaryotes diversified, we're going to find more formations with eukaryote fossils in them. That makes sense. And I was inspired by this paper, it came out a couple of years ago by Slater and Bolin, who looked at small carbonaceous fossil assemblages through the Tonian into the Cambrian. And so if you look at A there on the right, they have the number of formations in each of these time bins, Tonian, Cryogenian, et cetera. And then they look at the number of those formations that have animals, metazoans, or possible metazoans. And what's cool about this, and then on the right is they have the time scale instead. What's cool about this is is that you kind of see the appearance and radiation of animals through this time. You see nothing, none of them in the Tonian, Cryogenian, and then they show up and they become an increasing number of the assemblages. So we thought, oh, this is what we would expect to see if eukaryotes were diversifying through the Tonian. And so we approached this by asking, well, how many fossiliferous units are there through this time interval? So any fossils, and so this is, prokaryote-bearing assemblages. And what proportion of those through time have eukaryotes in them? And we broke them up by likely and possible eukaryotes. Again, it doesn't really seem to matter. So the cherts there are on the top, the shales are on the bottom. And I think it's important to note that we are dealing with a very small number of assemblages. Tina did this work. She scanned through the literature for fossiliferous units. So you can see, okay, it looks like there is a real increase here, but oh my gosh, we're going from 2 units to 4 units to 8 units. Is that really statistically significant? And so Tina did an analysis where she subsampled the data to generate these blue or orange areas to show the range of possible values that we would expect if there were no change in the frequency of eukaryote-bearing formations through time versus total fossiliferous formations, right? So it's basically sort of the null hypothesis are those windows. And if you're outside that window, it's telling you something, that there is a change. So here in the shales, you see really the only change is from the Orosirian into the Statherian. It sort of makes sense. We're sort of at the end of the beginning of the eukaryote record. But significantly, there is no evidence for a shift in the Tonian, so, with the exception of the appearance of VSMs. Okay, so the way to read this is there is no indication that the proportion of eukaryote-bearing formations versus all fossiliferous formations that we know of has shifted through time. And it's interesting here, this VSMs, that's a whole other story, but VSMs really do have show up in a significant way in the cherts in the late Tonian. Okay, so we say, we conclude that there is no evidence that Y is driving X, that this diversification results in more and more formations. Another possibility is this hidden variable idea, this common cause. And we were inspired by work that Shannon Peters had done in the 2000s looking at Phanerozoic diversity, marine invertebrate diversity. And he found a similar correlation as we do, sort of an R squared of like 0.6, suggests between sort of rock volume, number of formations, I think it was, and diversity through time. And he argues that that's real and that what is going on, so that's real in the sense that, the diversity pattern is real and the rock variation is real. And what's going on is that as sea level rose, we had greater areas of shallow shelf, these wide shallow seas where marine invertebrates could flourish. And there's a well-known relationship between habitat and species number. And so if you have a larger habitat, you have more species. And then time periods when the habitats were destroyed, diversity went down. So this is this common cause idea. And we think that is worth considering in our case. But I think the final possibility that there's a sampling bias is probably the most likely explanation, that X is driving Y, that the number of assemblages in our data set, the number of assemblages that have been studied is what's driving the diversity pattern of eukaryotes through the pre-cryogenian time. So this got us thinking about this model that I introduced at the beginning of the talk, of this delayed eukaryote diversification and the idea of a Tonian radiation. And I think it's important to note that if you go back through those papers, those reviews of early eukaryotes, that idea that delay is based primarily on the pattern of taxonomic diversity. That idea has its origin in these papers by Vidal and Knoll, and then Knoll, 1994. And then there were additional corroborating lines of evidence, but they were pretty weak, to be honest. So it's based on this biomarker pattern, which of course is really poorly sampled. And in fact, Brox has called into question this interpretation that it's reflecting a dramatic eukaryote diversification, suggesting that some of these older biomarker assemblages have been misinterpreted, and that in fact a lot of these could have been made by eukaryotes as well, and that there was this early world of stem group eukaryote diversity. Whether you like that idea or not, I think it's hard to disagree with the idea that the biomarker record is poorly sampled and is probably also affected by biases as well. The other line of evidence, these biological innovations, well, one of them is this apatitic scale microfossil, which is a single assemblage, and these fossils would be really easy to overlook. And so who knows when this group actually evolved? You can't say that, these are an indicator of a Tonian radiation. And then the last thing is those vase-shaped microfossils. Of course, this is a group that I have worked on. These are fossils that do appear pretty much all over the world around in the latest Tonian. And so I do think that they're signaling something, but whether you want to hang your hat on this idea that there was a Tonian radiation on the basis of just the VSMs, I think is a little bit of an overstatement. So we're left with this question of, is this pattern of a Tonian expansion of eukaryotes, is it real? And if it's not, then have all of our efforts to sort of try to answer what's going on in the Tonian. I mean, it's worthwhile to find that out anyway, right? But I don't know that there is necessarily something to explain in terms of a biological phenomenon, in particular in the Tonian. So I'm in this weird place where when this result came out, in my heart of hearts, I was like, well, of course there's a Tonian radiation. The Tonian radiation is real. But my head is saying, what's the evidence for a Tonian radiation? And honestly, like I can't really point to anything that convinces me. So where do we go from here? I mean, the result of this paper, I don't think that we have necessarily settled on a definite explanation of what's going on with this correlation. I think we're still left with these, three different possibilities. I should emphasize that like the work we did looking at, the percentage of eukaryote-bearing formations versus all fossiliferous formations, what we were really trying to get at, but we don't have data on, is sampling intensity by paleontologists. What we really want to know is how many units have paleontologists looked at and either found fossils or not found fossils, right? The problem is that paleontologists don't report when they don't find fossils. But it would be great to have that information, because you would expect that maybe if this were true, we would see the total number of units that paleontologists have studied, that eukaryote-bearing formations would be an increasing number of those, if this were true, that diversification were driving an increase. Common cause, I think that's worth investigating. Here, we would need to compare diversity or eukaryote-bearing formations with total sedimentary rock volume. And Shannon Peters, I've reached out to him to talk about this, to see if we could explore this. He has a data set for North America, and the goal there is just to expand that to other parts of the world. And then finally, this idea that I guess I favor, that sampling bias is really what's driving the pattern. Again, we need to get a sense of how intense our sampling has been and how that correlates with eukaryote diversity through time. So we need this same proxy, number of units sampled by paleontologists. So what's the takeaway? The first is that the pattern of eukaryote diversity has changed significantly over the last 50 years. And I think the fact that there has been such a big increase in the Mesoproterozoic and the Paleoproterozoic is probably telling us that sampling bias is what's driving this pattern, right? That it's so susceptible to that study. Diversity correlates extremely well, R squared greater than 0.9, with the number of eukaryote-bearing formations in the data set. We think maybe common cause could be driving it, but we think more likely it's sampling bias. So the overall takeaway is, I don't think we know what the pattern of eukaryote diversity was before 700 million years ago. And I think we are, if you think about a collector's curve, where with every new sample, you're increasing the number of species. And if you sample more and more, at some point you kind of saturate. I think we are maybe here on the Precambrian collector's curve. We are still basically just going straight up with further sampling. And if you go out and study a new formation, chances are you're going to get 1.24 new species per formation that you study based on this curve. This raw data, which I think this is fascinating, it shows us sort of where to target. So this is, I showed you this before, right? The monographs first pass. This is separated by these different 200 million year bins. So what this tells us is this 1900 to 1700 million year window, very poorly sampled. This 1300 to 1100 million year old window, interestingly, also quite poorly sampled and relatively low diversity compared to some of the other units. Cherts have almost no eukaryotes throughout most of this interval, and it's only in the Tonian that we start to see the significant number of units that have eukaryotes in them. You see the ASM is a big spike there, and a lot of this is due to the V-shaped macrofossils showing up in the cherts. What does this mean for the big picture of eukaryotes? This paper came out. This is a really nice piece of work by Tong et al. Using the CONOP approach to look at eukaryote diversity through this whole interval. And I'd say that their results pretty much, well, what they suggest is the real change here is across the cryogenian glaciations, that before this time, eukaryote diversity overall is quite low. And I think that is consistent with what we might find. I mean, I guess I'm arguing that we don't really know what the pattern is at all. But I suspect that probably there's not much of a, there's some oscillations through this time interval, but really the big change is across this interval. I think we should, instead of referring to this as the boring billion, we should maybe refer to it as the barely sampled billion. Anyway, so I think that's the end of what I want to say, and I welcome your questions and your feedback and ideas about where to go from here. Thank you.
Thanks a bunch, Susanna. It was really cool. I really like seeing everybody's different names for the boring billion. I think it's funny. Okay, so we'll switch to questions and discussion. You can raise your hand with the raise hand button or type it into the chat. The first one was Greg Vitalik. Please go ahead.
Oh, I was first. Okay. That was wonderful. I love it. You're pushing them further and further back, which I'm a big fan of because, well, I think I have some that further back. But it seems to me sampling bias has to be the answer. Because eukaryotes are basically aerobic creatures, but they're only preserved in anaerobic environments. And we have actually two other lines of evidence that the Tonian is a real thing in terms of eukaryote evolution. because of paleosols, because of the salts in the paleosols. We go from predominantly gypsic paleosols in the early Proterozoic to calcic paleosols starting to come in increasingly abundantly by the Tonian. And also, if you look at all those paleosols that were studied by Dick Holland and many others, the deeply weathered ones, we can actually figure out the rate of weathering of silicates in paleosols through time. And it also climbs right then. Not only the rate of cation depletion from the profiles, which we can get quantitatively, but also the rate of apatite depletion, which is phosphorus liberation from apatite. So I think what you need as a metric of this eukaryote flourishing is something else. And I think that does support the Tonian increase. And furthermore, I think that Tonian increase, particularly on land, created snowball Earth.
Well, I will tell you, I'll share some unpublished data with you, and it has to do with those vase-shaped microfossils. We have discovered a new assemblage. Well, Bonnie Bloser actually worked on it initially, but we've dug into it in more detail from the Chuar group, a little bit lower than the areas that I had studied. And these are extraordinarily well preserved, organic, walled, and they appear to have what look like scratch marks, which we think maybe could reflect aeolian transport. And they, we, for various reasons that I can't go into at the moment, don't have time, we suspect that maybe those vase-shaped macrofossils were in fact terrestrial or living, you know, sort of in whatever a Precambrian peat bog is like, near shore. And so I do wonder if something significant is going on starting 750 million years ago, maybe, at least with the fossil record where these vase-shaped macrofossils appear everywhere. And as I noted, you know, they're really the dominant pattern showing up in those cherts, as opposed to all the other eukaryotes in our data set, that I wonder if that is a signal about what's going on with life on land.
Absolutely. Yeah. Well, as you know, I had an ill-fated expedition into the Grand Canyon myself, and I found them in a paleosol, in a well-drained paleosol. And what stimulated me to take on that expedition in the first place is we know that these VSMs are almost only in soil today. They don't do well in aquatic environments at all. So from the very beginning, they were probably there. So your new my approach was.
Sorry, they're in aquatic, but just not marine. They're not common in marine. Yeah.
Yes.
Yeah. They're definitely in ponds.
And yes, if they're in cherts, they're probably in ponds. But mine were actually in the red paleosol itself, and it was a calcic paleosol, which that's the kind of rule in the Atacama Desert. When you go from gypsic to calcic, which you see both in Chuar, you're in the eukaryote territory. When you're in the gypsic, you're in prokaryote territory. And it's that balance of the salts that is another clue to what the eukaryotes are doing. Anyway, wow, I love it. Thank you.
All right, thanks. So next up, Jingjun, please go ahead.
Hey, Susanna, that was really awesome, eye-opening for me. I have many questions. I will ask one for now and then yield. You know, I'm not a paleontologist, so can you elaborate on why the apatite scale microfossil can be an artifact, given that in the phosphate record there is a number of increased occurrences in the Proterozoic?
Yeah, sorry, an artifact. They're not an artifact. It's more that they are so special that they are extraordinarily diverse, and they are exceptionally difficult to, I would say they would be exceptionally difficult to find. They would be very easy to overlook. They are just 10 microns or so, 10 to 20 microns in size. And, you know, typically when you're a paleontologist, you're looking through these slides and your eyes are more set on finding 100-micron size things. They're also sort of can be almost clear, you know, they're not sort of the dark brown that you expect to find with organic wild microfossils. So I think part of the problem is it's a search image issue. It could also be that they're rare. I think it's just at any time in paleontological diversity analyses, if you find something like a Lagerstätten, right, like an exceptional deposit that is just this incredible window on life, the species numbers can get really high. You don't want to take that seriously, right? You wouldn't want to say like, oh my gosh, there's this radiation of animals in the Middle Cambrian because you include the Burgess Shale, right? Like including the Burgess Shale is not in a diversity analysis is going to result in a peak, but you don't want to take that literally and be like, oh, what happened in the Middle Cambrian? There's a big diversification. It's just that we included this really special unit that is, you know, occurs very rarely. So that's why I sort of made a point of those apatitic scale microfossils. They're special, they're real, but I don't think they're telling us that, you know, they're only appearing in one place. So you can't say like, that's when the diversification happened, if that makes sense.
Yeah, it makes sense. Thank you.
Yeah.
Thanks, Jing. Prescott is up next. Please go ahead.
Dr. Porter, this is really cool and a really neat new perspective on all this. I was curious, when you're talking about the potential causes for the relationship that we're looking at and talked about either the sampling, like what we're actually looking, or looking at the total number of deposits. I was curious how taphonomy could play a role in the observed record, particularly just considering that, at least maybe from like a Phanerozoic perspective, every deposit where we're finding these fossils is a deposit of exceptional preservation, and that we're very likely not getting a full representation of everything that was there because it requires such unique circumstances to even preserve the couple of species that we do find in any given deposit, and how, I guess, acknowledging that, and then how might we deal with that when we're trying to develop a more comprehensive understanding of how this is changing through time?
Yeah, that's a good question. So Tina Woltz published a study, and it was building on some thinking from Nick Butterfield and others earlier that looked at this phenomenon that we see there seems to be a relationship between total organic carbon in shales, which is really the predominant record of early eukaryotes is in shales. So TOC and preservation. And Tina showed that there does seem to be a significant relationship between those two. So if you have TOC less than about 1.5%, that's where you're going to find the really well-preserved organic wild microfossils. And then as that goes higher, you're going to see more poorly preserved and then nothing. I think whether that's sort of an ecological signal, like they're, you know, has to do with the habitat that they're living in, or partly also contributed by, you know, actual preservational conditions. It may be a mix of the two, why that's happening. But it's easy to think then, to wonder, you know, yeah, the extent to which, you know, if we were to intensely study, we've known about this relationship. I mean, I know it's been like a rule of thumb that like Andy Knoll told me about, and, you know, Nick Butterfield and Shuhai have used this sort of rule of thumb. Don't target black rocks. Don't look for organic rich rocks. Look for gray or green shales. But you could imagine if intensity of sampling didn't pay attention to that, and, you know, we were intensively sampling organic rich rocks and finding nothing and nothing over and over again, that could, to think this through, that is certainly going to influence the diversity pattern. So sampling, paleontologists have known this for a long time, obviously. Sampling affects diversity patterns. These taphonomic megabiases through time affect diversity patterns. And we have to be careful thinking about that as well. I think that our study, though, maybe because the R squared is so high, it really, I mean, there's no escaping the fact that graph is telling us that basically Leanne didn't have to go through all that effort to identify all those species, right? All she had to do was just count all we had to do is just count all the formations that have eukaryotes in them, and we can use that to plot diversity through time. I mean, I think that, I think probably sampling is the dominant control, but it's an interesting question to think about, that phenomenon too of like the nature of paleontological sampling, where we have this first pass and then we go back and study and we find more and more. I don't know how to quantify that effect.
Cool, thanks.
All right, thanks, Prescott. There's no more hands. You have some stuff in the chat, some great talks, and thanks. Eric Sperling said, I like seeing the note on sampling bias and sampling interval applied to the biomarker record as well. Issues of sampling also affect the geochemical record just as much as I think the geochemical community can take a cue from the paleontological community and continue to think more deeply about all the different all the different biases that affect the record we see.
Yeah.
And that's more just a comment. And then Vladi Alterman says, is that the right approach? Only because the Burgess Lagerstätten is so special, it doesn't mean that animals were not common at that time. They were just not preserved in other conditions, or the right conditions, or the right conditions were rare. But the species might have been common and widespread and diverser than we see from the fossil record. And then wouldn't it be logical to expect radiation with time?
Well, so to address the first question, I think everything you're saying is right. That with fossils, we're only seeing a fraction of the species that were present at that time. It's more of an issue of apples and apples and oranges that we want to, ideally you would want to sort of look through the same preservational window through time and look at this sort of the same environment, et cetera, and see how diversity is changing. So it's not an issue of, it's not an issue of those things that you're saying. It's just a matter of, you wouldn't want to infer that, you wouldn't want to mistake a moment of exceptional preservation for an actual change in global diversity. Those are two different issues, if that makes sense. Would it be logical to expect radiation with time? Well, there have been periods of time when diversity has dropped. So I guess it's probably logical to think that, over time there was an increase in diversity. But the question is, when are there big shifts in eukaryote diversity through time, as the way it's been represented with the Tonian being sort of a flowering of a radiation of eukaryotes? Was it in the Tonian? Was it in the Paleoproterozoic? Was it in the Mesoproterozoic? Was it a significant uptick in diversity, or was it a slow growth in diversity? Were there intervals of time where it went back down again? I think the fact that it's so strongly correlated with sampling just suggests that we don't know what the pattern is. I hope I sufficiently answered that question.
I wonder if I could just jump in. This is Nick Butterfield. I can't see the raise a hand symbol. So I'm jumping in if that's all right.
Okay.
Thanks for that. That was really interesting, Susanna. I've a question. What you've presented is are issues about total diversity. These are raw taxonomic counts, so we can worry what a species is, of course, and different practices for how you derive species. But nonetheless, what you've got are raw counts. Now there is a potential for substantial turnover.
Yes.
And there are definitely new kinds of things turning up in the Tonian. And that hasn't gone in to your calculation. All you're telling me is raw counts. Well, if you look at the Paleozoic, there's nothing going on in the Paleozoic as far as diversity is concerned. It's just that plateau, right? But there's zillions of things going on, and I'm just wondering, aren't you missing something here?
Yeah. Oh, probably. I'm sure. Yeah, I'm sure. And that's a great point. There does. I mean, if you look at an early Tonian or late Tonian organic walled microfossil assemblage, there are some of the same species that we find in the Mesoproterozoic, Paleoproterozoic, but there's a lot of new species. And you're right. So turnover is absolutely something that is fascinating to think about and not something that we have looked at with our data set. But that's different than global diversity, right? And so it could be the case, say, that there is turnover. I think there is turnover into the Tonian sometime there. I definitely think I think there's turnover associated with the cryogenic glaciations. But whether that is a radiation, right, that needs explaining by an increase in oxygen or an increase in nutrients, I don't think so. I mean, turnovers are usually associated with extinctions, right?
All right. Thanks, Nick. For those struggling to find the raise hand button, just look at the bottom of your screen. You'll find the reactions button. It has a heart there, and then you click that, and the first button in the pop-up is the raise hand. Leanne, please go ahead.
Hi. So I was just going to kind of chime in, if I could, on the apatitic scale microfossil issue. And kind of part of the reason that the apatitic scale microfossils are so difficult for us to deal with is, well, kind of two reasons that I'll break out right now. So because they're only seen in the one unit, we don't have a good sense of when they actually first appeared and then their last occurrence. So it's hard to actually say anything about, them in terms of when they first show up, because maybe they were around in the Mesoproterozoic and we just don't know it. And because we have only the one unit, we have low confidence that we understand when they actually first appeared. And then secondly, and probably more importantly, we see lots of different morphotypes. So they're broken out into lots of different species, but we don't have any confidence about how many actual biological species that means. Because with a lot of modern day plate bearing or scale bearing organisms, you'll have one organism that will make 3 or 5 different morphotypes of scale on its body at any one time, excuse me. And so just with those things in mind, we wanted to understand that the ASMs needed to be treated carefully. So just to chime in on my 2 cents on the ASMs and why they are treated differently in this paper.
Yeah. But yeah, thank you, Leanne. I forgot to make that point. And that's something that I think Nick and Shuhai also raised as reviewers, that, thinking about some of these fossils, including the organic walled microfossils, could be parts of a larger organism. So, a single species is being counted more than once. But I think the bigger picture takeaway, right, is even when we included the ASMs, the R squared went up, right? It was 0.99. So really, as with, all that work we went into parsing likely eukaryotes versus possible eukaryotes, doesn't matter. the signal is so strong that there is a correlation between the number of formations of any given time and the number of species of eukaryotes.
All right. Jingjun and Prescott, you guys have both asked a question already, so I'm going to hit ones that haven't, and then we'll come back to you guys. So in the chat, Sana Mughal, I'm sorry if I mispronounced your name. Hi, Susanna. Amazing talk. Did you look at diversity at higher taxonomic ranks, though I know this is limited? And how do you think poor taxonomic controls plays into inflation of genus and species towards the Ediacaran?
Yeah. So the problem with looking at diversity at higher ranks is that, yeah, as you say, it is so limited. So I could say, okay, there's vase-shaped macrofossils, that group shows up. It's just, I think any attempt to parse the organic walled microfossils into different higher taxa, it's I would feel uncomfortable about grouping very many of those. Like I think it would just be artificial. So I didn't show this, but we did look at multicellular groups, and that's something that actually also had been suggested to be another line of evidence in support of Tonian radiation, is that we have more types of multicellular cenocytic forms in the Tonian, but of course that also, not surprisingly, would be affected by sampling, right? But if you look at that curve, yeah, I mean, there's just so few of them that I think it would not be worthwhile to spend too much time thinking about the higher taxonomic ranks. Interesting thought, but yeah.
All right, thanks. Jingjun, I'm guessing your question was covered in Leanne's question?
No, it was I have new questions.
Okay, so Prescott, please go ahead.
Yeah, so I was wondering, you mentioned this when you're talking about the that science has this problem with not publishing negative results or the lack of results. And I think in particular, your work here highlights an additional reason to maybe the more cited reason of not wasting resources repeating the same science, even though replication is crucial to science. But I was curious if you had any ideas or suggestions or recommendations about how we might be able to kind of, as a community, either in our little subdivision of paleontology or in paleontology overall, how we might address this problem when this not finding fossils is super important to our understanding of the fossil record, like how we might go about making that data available.
Yeah. Well, so we've tried to do this. I mean, so Leanne, for example, wrote a paper about a number of different drill cores that she studied, looked for fossils through the Cryogenian. And one of them was entirely barren. But she included that and reported those barren samples probably in the supporting online material, you know. But I think maybe that's a way to do it. I mean, if you obviously find nothing, that's yeah, I guess there's a way to spin that and publish it. I can see how you wouldn't want to put a lot of effort into it. But I think as part of, you know, broader paleontological analyses, if you study a bunch of drill cores or you do a bunch, you know, you sampling of a unit, like probably you're going to find Leiosphaerids. But it would also be really helpful if you reported all of the samples that were barren. And I think I'm finding that maybe more and more that in more recent paleontological analyses, there will be, you know, a spreadsheet that will report this sample was looked at and here's the species I found, and this sample was looked at and it was barren. I think that's sort of thinking about where to go next, like a proxy for paleontological sampling intensity through time. I'd love to do. And the only way I can think of is to maybe just focus on those papers that actually include those data, barren samples, fossiliferous samples, barren samples, and then use that. No, I mean, journal, what's that? The journal of, not your, isn't there a journal out there? Like the journal of not finding anything.
Sounds like a tough proposal to get funded.
I know, yeah. Exactly.
Yeah.
Totally.
But really important. So.
It is, yeah.
All right, Jingjin, go ahead.
Sorry, me again. So I have a question about the one of your figures shows the species and formation over time. There's a interesting plateau in the mid-Proterozoic, and I wonder is that just from the black shale, or is just in all other rock you have this pattern?
Yeah, so that's something that is really interesting, and it actually didn't show up even so well in the data that I presented to you because of the bin sizes. But there is an interval in the mid-Mesoproterozoic, like 11 to 1300 million years ago, where there's almost nothing. No eukaryotes from that interval. And no studies, right? And so I have to think that there's something interesting going on geologically. And I see Paul Hoffman has his hand up. I mean, maybe the geologists in the audience can comment on that. I think it seems to broadly correspond to supercontinent cycles, that maybe this is just an interval where there aren't a lot of rocks, or there are rocks that we just haven't dated, There's a lot of many I should have said this, like there are some assemblages where we just don't know the age. And so it's hard to know how to include them in this analysis. Yeah, there's definitely interesting. I mean, that jumped out to me, right? That the early Mesoproterozoic, late Paleoproterozoic, we actually have a pretty rich fossil record from that time. You saw that big increase relative to the Knoll 1994. That is sort of almost getting in the same realm of the diversity we have in the Tonian. So I think that, yeah, there's some signal going on there with rock volume, I suspect. Or, what countries and continents we Precambrian paleontologists come from and what we're able to study. Right.
All right, thanks, Jingjin. Go ahead, Paul, please.
It's hard for me to believe that the scale microfossils are a unique occurrence.
Right.
And also, it's hard for me to believe that a much smaller number of species are making so many morphotypes, because compared to Phanerozoic scale microfossils, these Tonian ones are enormous in size.
Yeah.
So my question is, are we looking in the right way for these fossils? In other words, are we doing what conodont people do, which is dissolving large volumes of limestone in weak acid to look for these apatitic scales? Are we looking for them in the right way? Because as far as I know, and I'm not a paleontologist, this is not the normal way Precambrian people look for fossils.
Yeah, exactly. Thanks for bringing that up. I think that's a really important point, that the way Precambrian paleontologists have traditionally looked for fossils is in mudstones and in cherts, in silicified carbonates. And if you're just presented with a carbonate, not silicified, I've been out in the field, I get carbonates, I often don't sample them. And I think that was a really interesting thing with the cryogenian, with Tanya Bosak's work and Phoebe Cohen, who thought, oh, well, I'll go ahead and sample these carbon, I'll dissolve up these carbonates. And that was a real surprise to me, like Phoebe found those macroscopic organic warty sheet fossils, which are fascinating, and they're preserved in carbonate. And it never would occur to me to look for organic fossils in, so that's an example of organic fossils. I agree with the scales. Like, I think it would be great to do that, but you know, I think there are many fewer Precambrian paleontologists than conodont. Maybe not anymore. Maybe we outnumber the conodont workers. But there's, you know, a vast amount of rock and not a lot of Precambrian paleontologists studying.
I presume Phoebe sampled that location because Allison had described fossils from there.
Yes, right. Yeah. And the story with Allison, Carol Allison, is that she had bad eyesight and had to turn the microscope light down really low, and that's how she was able to see these scales that otherwise, if you were just looking quickly through it, you wouldn't notice them.
Yeah, hers were in chert, I believe.
Yeah, hers were in chert, absolutely. There's also the issue of a search image, right? Like, yeah, I think a lot of times, you know, we go into these searches with a particular idea in our mind about what these fossils should look like, and if it doesn't fit that, we don't see them.
We should ask Phoebe why she collected the limestone samples.
I don't know if she's here. My understanding is that when she and Nick was part of the study, when they found that those fossils were apatite, I guess it's true if they were silica too, they were like, oh, well, they should be in the carbonate that are right outside. And that motivated, it was based on knowing what their composition was, and thinking that, oh, they should be in the carbonate right outside.
Yeah, but they didn't know that. She didn't know that when she was first there. And I think she was only there once.
Oh, that's right. Well, she
somebody
thought
collected 5 or 6, limestone samples.
Right.
Yeah.
Well, and you can't not collect the limestone if you're collecting. I mean, the chert nodules are super tiny in the limestones.
Anyway, I think there's got to be a record of scale microfossils, much more extensive than we have the inkling of.
Yeah, absolutely.
So paleontologists need to become more pioneering and sample formations that haven't previously yielded fossils.
Yes. Yes. Give us funding.
Mm-hmm. All right, Jinjin, ready to weigh in? Go ahead.
Sorry, this is about my last question. So this is more kind of generic question related to my own research. For non-paleontologists, do we know like the partitioning for all the Precambrian eukaryote fossils, that proportion that are the habitat is on land versus on the ocean? Yeah, do we have such database for that?
So other people are welcome to chime in here. I think there are a number of units that have been suggested to be terrestrial or lacustrine. And so the Nonsuch Formation in Michigan is one example. It's about 1 billion years old. And then the Torridonian Rock in Northwest Scotland they're another example. Those both have fossils in them. The Chuar group, where I've worked, there's been a lot of conversation back and forth about whether those assemblages are marine or lacustrine or, mix of both. So I think it's not always easy to be able to know for sure that you're dealing with a lacustrine versus a marine unit. There are paleosalinity proxies that have been developed that I think a lot of people are probably going to go out and apply them and see if we can maybe nail it down for a lot of these units. But even taking those examples, the fact that I can sort of list them, gives you a sense of how rare they are relative to the other units. But I'd say even the other units, I mean, in all cases, we're looking at, for the most part, like shallow seas on the continents. But is there a motivation for your question? Like just a sense of, yeah, what terrestrial life is like?
Yeah, so I recently have a paper on the late stabilization of Earth's ozone layer. We found that no matter how high your oxygen is, you cannot stabilize your ozone layer to provide the full UV screen until the Cambrian. Yeah,
Oh,
so that's the motivation.
Yeah.
Or the Devonian, according to iodate.
Yeah.
The kind of uncertainty on the final stabilization is like 300 million years. I don't want to get into that. We're talking about eon level changes. Sorry, Paul.
I think that the vase-shaped macrofossils might actually be terrestrial, you know, in the sense of like in ponds near marine margins. Yeah, it can be kind of hard to show, I guess.
I'd like to jump in here.
Yeah, go ahead.
I think eukaryotes were actually evolved on land and were much more abundant on land way earlier than all of this record. We have them back to 2.1 billion years in the Heckport Paleosol, the Paleosol that everybody loves and everyone and his dog has studied. It appears to be some sort of fungus. We also have these lovely slime molds, dictyostelid slime molds, trace fossils that Stephen Bankston studied in Western Australia at 1.9 billion years, again, in paleosols. The nice thing about paleosols is, well, they're preserved regardless. They don't always have fossils, and we can tell whether they're likely to have fossils or not from their redox state and from their pH, which we can also estimate chemically, and from the mineral content. The only thing that controls the distribution of paleosols is whether they're eroded away or elided in some sort of disconformity or something, but their record's as good as the formation record, really. So yeah, in answer to Yinggun's question, I think life was on land very early. As far as the UV flux goes, Carl Sagan and many others showed years ago, those little microbes, they can hide behind a single quartz grain and be very happy photosynthesizing in a very high UV flux. I don't think that sterilizes the land at all. Enough of my rant.
That's an interesting question, right? I wonder if we could get at it from a genetic, genomic analysis viewpoint, is seeing if there are.
Oh yeah, the Merkel clocks all show these things going way back.
All right, so we don't have anything else in the chat or hands raised. I'll give a second in case someone wants to chime in. All right, let's thank Susanna. Thanks for the presentation and the really good discussion at the end.
Yeah, thank you so much. I really appreciate your questions.
Fantastic, thank you.
Yep, thanks a lot.
All right, thanks everyone. Take care. We'll see you next week.
Thanks, Susanna.
Thanks, Paul.